Hello, everyone. Welcome to this Australian Biocommons webinar. My name is Georgia, and I'm the Australian Biocommons BioCloud Training Manager, and I will be your host for today. In this series, we share useful information about the latest digital techniques, data, and tools for the life science community. Each month, we hear from local and international experts who present a bioinformatics topic that will support Australians to deliver their best environmental, agricultural, and medical research. You can keep up to date with the latest Australian Biocommons news and events via the channels listed on the screen. Before we begin, we'd like to take a moment to acknowledge the traditional owners and their custodianships of the lands on which we meet today. In my case, this is the Gadigal people. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognize their valuable contributions to Australian and global society. Today, we're thrilled to welcome Dr. Sarah Beecroft to speak to us about how to use containers in a Nextflow workflow. Sarah obtained a PhD in medicine from the University of Western Australia in 2020. She has a background in both laboratory and computational genetics, and she is now part of the Supercomputing applications team at Pawsey, working to support bioinformatics users through workflow development and optimization, training, and advocacy. Welcome to the webinar, Sarah, and I'll now hand it over to you to start the presentation.
Awesome, thanks. Thanks for the intro. Yeah, thank you for joining this webinar. I hope you'll find it useful. The more I learn about Nextflow, the more I realize how much I don't know. There's so much to learn, and same with containers. So the aim for this session is to give you a bit of a refresher. If you've come across these things before, a bit of a refresher, and if you are new to these concepts, it will start relatively from the bottom, and we'll build our way up. So let's get started. So why do we need containers? Well, I would say that bioinformatics has a bit of a reproducibility problem. I know I've certainly had this thing of like, it worked on my laptop or it worked on this system, and now you can't produce the figures from your 2023 paper. You can't install any of the tools. Or why does the same pipeline give different variant calls on, Supercomputing versus our old node? Or how can you satisfy reviewer 2 with your software stack so that you can get your paper published? These things can take up a huge amount of your time, and if you're in research, it's taking time away from your science because you need to be doing all of this software administration. So hopefully you'll be able to save some time on this stuff so you can do more interesting things. So just a bit of a briefing on what we're going to be looking at today. So why containers? Why are they useful? How do they work? Using Singularity on HPC. So why aren't we using Docker? And just a quick run through on the most important commands. Some talking about the interconnect between Nextflow and containers. And if you're really into Nextflow and containers, there's some extra info for the deep divers out there. And also a working config that works on Zatonics. So you can see a working example of type of thing you can use. So I saw this image quite a while ago, but it has really stuck with me of like, how hard could it be to reproduce a pipeline? I'll just download the things and it'll just work. I've certainly been there, and it's more difficult than it should be, I think. And definitely step 6, try not to cry. Cry anyway. This, I think, can really sum up the experience. Because we do have a lot of moving parts in bioinformatics. There's a lot of complexity in the tools that we use. There's lots of different versions, and the documentation isn't always fantastic. I've realized in other domains, which I come across working at Pawsey, they have perhaps one piece of software that does everything that they need, which is absolutely crazy to me. So you only need to become an expert in one thing, whereas for us, we've got to be across all sorts of different dependencies. So part of the problem, I would say, is not only you have to be across lots of different tools, but also you have to be very aware of the versions of things. So for example, if you're getting using sometimes a different version of one tool can give you actually a meaningful difference in your scientific output. So you need to be extremely careful about what versions of things you're using and documenting that. Because if you're looking 6 months down the line of like, oh, that's really weird. I analyzed this person's genetic data at this time point, I got X result, and now I've analyzed it 6 months later, I'm getting Y result. Why is that, and what's biologically meaningful versus just artifact or noise? Well, you need to be able to pin that down. So understanding what versions you've used and being able to reproduce those can be extremely important. Containers can help you with that, because once they're built and you have the container in your system, it's immutable essentially. So you can know exactly what you've used. Also, using pre-built containers can save you a lot of time with installation pain. I remember during my PhD, I spent, excuse me, I don't know how many weeks trying to install VEP with a lot of these Perl modules, and it was just absolutely terrible. And now you can use a container with everything pre-installed inside it, and it'll save you quite a lot of work. Containers are also useful in that you can, in general, run them, for example, on your local Linux box under the desk, in the lab, or on a cluster, or your collaborator in Switzerland can run it on their cluster. So it really helps if you're working across a lot of different systems, or you need to run your stuff or like share your workloads across different systems. It also helps you do a bit more of an audit trail. So it's not necessarily completely perfect using containers, but it's a lot easier to have these things pinned down, particularly if you're very particular about using a hash rather than a tag. But we'll talk about that as we go. So here's a bit of a breakdown of key concepts around containers. What are they and how do they work? So when you have a regular installation, for example, on the left here, let's say that these are all different tools that we want to install. Typically, if you're just installing them bare metal, as you might call it, straight onto the system, all of these tools will be sharing libraries and dependencies. And this can very quickly become dependency hell, because what if you have tool A needs a certain set of dependencies and tool B needs a different conflicting set? Well, then you're in trouble because you can't have both installed at the same time. Whereas if you have a container, the libraries and dependencies are isolated. So they're not interfering with each other. You can have as many different libraries and dependencies as you want. They're not touching or fighting. And you interface with that through this layer, a container engine, as opposed to going direct, like in the system on the left. So in short, a container contains everything that your software needs. So the libraries and dependencies, basic OS, install commands, environment variables. It's all in there. So all you need to do is hit run, basically. And so when you do this, you don't just speak directly to the container as such. You need to go through this middle layer called a container engine. And so there's a bunch of these around. It's a type of software. And so you, as the user, you issue your command to Singularity, is the one we'll be talking about today, and it gets the container to execute the task that you want and give you some science or whatever else that you're looking to do. So the two main ones I would say are Singularity and Docker. Docker was the first one, and it's become kind of the de facto standard for like how you write your containers, how you create the recipes for them. But Singularity is more formulated for use on HPC. So in terms of having what container engine will you use? Most places would use Singularity. And the reason for this is that the Docker engine was designed for cloud computing originally, and it needs sudo access for a lot of these things, which obviously you don't have on a HPC system. I don't have sudo access myself, and I work there, which is for the best. I'm very happy that I don't, because with great power comes great responsibility. So with Singularity, it runs as you. There's no root, there's no daemon. The other useful thing with Singularity is that the container becomes a single .zip file that you can move around, so you can copy or move this file, which is quite easy. And it also will auto bind mount, home, your present working directory, temp, these types of things. So the bottom line is Singularity was designed for unprivileged users on shared file systems, and that's us. And so, as I was sort of alluding to, you can use Singularity and Docker to also create containers. So you can run containers with them, but you also use these tools to create the containers if you're writing your own, which most of the time you don't have to, but you can if you want or need to. But the key thing to keep in mind is that if you're using these different tools to write containers, they have different syntax for writing the container recipes. And they have a different file extension. So with Singularity, this is .zip, which is mainly what you'd be using. So if you are writing your own containers, you would typically write them in Docker format. And then if you want to run it with Singularity, you pull the container from the internet, even if it's written in Docker, that's fine. In Singularity, we'll do the conversion to a .zip file on the fly. And run it for you. So that's kind of the standard setup, I would say. Singularity can also obviously run its Singularity format containers. Like, if you've written it in Singularity format, you can run it, but it's just not as good writing in Singularity format. So Docker syntax for writing containers, Singularity for running them is the way to go, I would say. So something that you might not know about if you haven't used containers a lot is this resource called Biocontainers. So you don't need to actually write your own containers most of the time. They're already out there. And Biocontainers is a great source for these because they've automated builds of all the Bioconda recipes. So it's one tool per image, and it's indexed by version and by hash. So you can look on biocontainers.pro. They're also on key.io, which is another repository where you can find containers and grab them, and Docker Hub. And so you can pin the version if you want. So for example, if I want SAMTools 1.2 whatever, I've got a fairly good idea that it's this one and I know what I'm getting. But if you really, really want to be very clear, it's better to cite the digest. So this is almost like a checksum, which doesn't change. So for example, with this version, the developer might push a slightly different container with that same tag, with that same version number to the internet. And so when you pull it down again, it's actually slightly different. Whereas if you cite the digest, you'll know if something has changed. So that's the most reproducible way. So Singularity in 60 seconds, there's a few key commands that you'll want to know. So first of all, how do you get a container from the internet to your cluster or to your Linux box under the desk? The main one, it would be Singularity pull. And so if you want to pull containers from the internet, you preface it with this Docker so that it knows that it's a Docker recipe and it needs to do the conversion to Singularity format. And you give it the address and the tag. And if you're looking on key.io, you'll see, how do you pull these things? Where do you find the address? You will find it on there. Once you have your container on your system, it'll exist as this .zip file. And so if you want to run a command using the container or inside the container, you would use singularity exec. So in this example, it's singularity exec container name and the tool, the command I want to run. So sound tools view, whatever. Sometimes you might want to open a shell session inside the container. And this is really useful if you're like trying to debug something, or you're using a container that's not from, BioTools or something like this. It's something that someone else has written, and you're like, oh, where did they put the binary? Where did they put this? What have they done? If you do Singularity Shell, you're inside the container. You can see all of the files and everything that are contained inside there and poke around. It's a little bit easier. So mainly for debugging. You can also use Singularity Inspect if you want to understand some of the labels and the metadata that are inside. So Singularity Inspect would print out a bunch of information that's held inside the container, which can be useful. So this could be a bit of a gotcha with using containers. So by default, because it's a feature and a gotcha, I suppose, you have a lot of isolation from the container is quite isolated in many ways from the operating system and from the host, as you would call it, from the computer you're running on. And so the default behavior is that you can't see directories outside of the PWD and whatever else has been auto-mounted, so like, home, various other things. So if I am running something and I like, oh, actually, I also need the container to be able to look in some very obscure directory that's not automatically mounted for me. I need to mount it myself. I need to make it available myself. And that's called bind mounting. And so this enables read-write access to these additional selected directories. So it's basically saying, I want you to also be able to read this thing, this directory. And you would do that with the singularity exec -- bind. So for example, if I'm, wanting it to look in this hypothetical reference directory for, some data, you bind it and then you give the container name and you can view now or look at the data that's over there. But you only have to do that if you're using pointing to a directory that's not already bind mounted. But it's an important concept to be aware of because it can catch you out if you're not expecting it. All right, so that was containers, a brief rundown. Now it's a bit of a refresher on Nextflow config optimization. Kind of stripping right back, going back to some basics. How does Nextflow think about your pipeline? Well, often with bioinformatics, you don't just have one process that you need to run, you have a sequence of processes that you need to do. And so Nextflow thinks about this as a graph of independent tasks. You describe the graph or the workflow, and Nextflow runs the thing. And so in this, example workflow, let's say you have some input files, you've got some fast queues, and you want to do something to them. Well, that's good. With Nextflow, you have your input, and it goes through something called a channel. So this is what kind of pipes the data between your various tasks. You have a process which does the work. So maybe you're doing some alignment with BWA mem. Cool. And then the output from that would also go through a channel and get some kind of final output, right? In reality, of course, you have many of these process steps, but just kind of getting these keywords down. So a process is one task. So for example, you might be running BWA, then maybe you run FastQC or whatever, and so each of these processes is self-contained. It knows its input, it knows its outputs, it knows its commands. And the data channel is what pipes the data between these processes. and it won't start a process until its channels have arrived. So it needs to know, oh, okay, the fast queue is ready for me. I'm going to do step A. Step A is finished, and I have the data that I'm looking for. Now I can start step B. And the workflow is the order of the processes and how the data flows between them. And so one of the great things about Nextflow is rather than, you could put all of this in, say, for example, a bash script, right? That's how everyone, I think, started out or starts out. You're like, all right, I'm going to just script it all myself. But then what happens if you have one of your inputs fail? Are you going to know? Do you have to run the whole thing again? Like, oh, so much overhead. Whereas with Nextflow, it handles all the retries, it handles checking what steps have already run successfully or not. It handles the parallelism in terms of running many jobs at the same time or many processes at the same time. And also can interface with Slurm or PBS or whatever, so that it handles all the scheduling for you too. So if you're doing a lot of these production runs, so you have a lot of samples to get through with this type of complexity, it can be a real lifesaver. So at a very simplified level, you have kind of two key files with Nextflow. There's the main.nf and the Nextflow config. And so the key concept here is that you're separating the what, like the workflow, the steps, the processes, and the how. And so by separating those, it means that you have a lot more flexibility in how your pipeline, works, in where it can run, if you want to scale it up or scale it down, rather than having everything hard coded into the what part. So the main.nf this defines the workflow, the data, how the data flows, what the processes are, the order of process execution. And so it specifies what are these processes, the commands, the inputs, the outputs we just talked about, right? And the idea is that it is portable. So nothing here mentions cluster specific settings generally, or it shouldn't. And historically, way back when, I don't know, like 5 years ago, it's ancient in bioinformatics terms, you did only have basically one main NF and one Nextflow config. It's gotten more complicated now, and we'll talk about that, but this is kind of the history of where it came from. And so with the config files, it says where the process should run. So is it, on your laptop? Is it on Slurm? You could even run things on like AWS or Azure or these types of places. And it says, with what does your process run? So is it using a container? Is it using a Conda environment? Is it expecting you to have these tools installed on your local environment, maybe if you're running on a laptop? It also says how much. So this is particularly important, if you're interfacing with a scheduler, for example. So you say, I want this many cores or this much memory or this much wall time or this many retry attempts upon failure for each of these processes. So yeah, this is what gives you that flexibility that you can take your workflow and run it elsewhere, because you can say, oh, well, today I want to run on Slurm, so I'm going to use the Slurm settings, but tomorrow I am going to run it on AWS batch, so I'm going to use the AWS settings. And next week I want to run it on my laptop, so I'll use those settings at that time. And you haven't changed anything in the main NF. So with the config, you can do a lot with these. And you can also mix and match them. So you can have more than one config at a time, and that's quite common these days. And you can also override existing configs with your own custom config. So, if you're using a pipeline that's been published, and that's typically how it goes, but you need to change some of the settings for yourself to run wherever you're running, you can do that. So the config is written in Groovy syntax, and Nextflow will read it on every run. So it controls, I feel like I'm repeating myself a bit here, but the executor, the queues, the resources, which container engine do you want to use, so Singularity, Docker, or whatever, where all of your CIF files are being downloaded if you're using those, per process specifications, and like the parameter inputs, this type of thing. So there's an order of priority. So the built-in defaults are the lowest priority. So if you don't provide any additional specifications, that's what will get used. But if you have a, just a general user level Nextflow config that's living in your home, this will override the built-in defaults from the pipeline. The pipeline might also have its own nextflow.config or, multiple types of configs that will override your local. And then probably the most important useful thing is you on the command line. So when you're doing your Nextflow command, Nextflow run, blah, 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 you can specify one or more of your own custom configs if you want or need to, which, can give you a lot of flexibility. You can also use these things called profiles. And so this is what I was sort of talking about before. We are like, all right, well, I'm using my pipeline quite a lot and I'm using it in many places. A really handy way to handle that is using profiles. So you can have your Sloan profile, your cloud profile, your local profile, all in one file, which is quite handy. And so with this, you can, keep all of this in one file. And you pick at launch which one you want to use. So you can say, for example, I'm running on Photonics today, I'm going to use dash profile Sloan. Or you might say, actually, I want to use Singularity in Sloan, which can be, very useful and a good thing to keep in your back pocket. So that's an alternative to having like many different config files, right? So you could have like a Photonics.config, a Singularity.config, a something else.config, or you can have them as profiles, which is a bit of more elegant solution. And I guess this is getting the point across that you can mix and match these as well. So you might have a Docker profile, Slurm, Singularity, and a lot of institutions now also provide their own profile. So Pawsey has one for Photonics, NCI has one. If you're running on, some cluster overseas, they, many of them have their own profiles, which you can find online, which can be quite handy. And so this is like a sort of bare minimum profile for enabling Singularity. So with this, it's saying, hey, Singularity is enabled. I want you to do these auto mounts so that it auto mounts like PWD, excuse me, home, temp, scratch, these types of things, which is typically what you do want. And on Photonics, for example, if you want to use Singularity, it's available as a module. You need to make sure that that's loaded. Otherwise, Nextflow will say, I can't find Singularity. And so you can make sure that's loaded in the background. And so if I enable this profile, it will let me use Singularity. It'll auto mount for me, which is really handy, and it'll load the module in the background. So I don't have to think about it. All I have to do is profile Singularity, which is pretty nice. You can also have these optional extras, which you might not need. It's up to you. Depending on where you're running, you might find that these things are super useful. So Nextflow will pull the containers from the internet for you on the fly, but sometimes, it can take a long time. You might want to have a timeout set. You might want to specify your own cache directory. So, you can put it somewhere that's more useful for you. You can also put in some run options. So for example, if I know ahead of time I need all of my containers to bind my references directory, for example, that's not in the auto-mounted list, you can also do that. So you can put all sorts of different run options and that type of thing in here. So there's also some environment variables that are worth knowing. You might not need to override these. A good sysadmin should have configured these things for you, but sometimes you want or need to do something different, and so you can set these. So if you want to change where to singularity pull all the CIF files and cache them, that's this variable. There's also Singularity's own cache, which is separate to the Nextflow cache. So when Singularity is pulling images, it writes all these like bits and pieces to this cache directory, tempter, and also Nextflow offline. So this stops Nextflow from checking online or compute nodes that don't have internet. So on Photonics, all of the nodes are connected to the internet, and you can therefore pull containers on the fly. But many HPCs don't have that, and so you would maybe want to use that setting. And so you can export these in your run script or have them in your bash RC, and they'll get read at runtime. So just going to pause for a little drink here, talking so much. So integrating with Nextflow. So I guess this is the more kind of advanced usage, getting these things to work together and changing things for your own use. So in general, you'll have one container per process. I mean, some processes might, use the same container, but you need to specify, what processes will get which containers. So in general, it's one container per process. Like you can't have multiple containers per process, really. So for example, if I want to do FastQC, it's going to use this FastQC container. If I want to do BWA, it's going to use this one. MOSDEF is going to use this other container. So Nextflow, you specify the container and where it lives on the internet, and it'll pull it for you, it'll execute it for you, it'll handle the bind mounts as we discussed. And you don't need to put in any singularity syntax in your commands. It'll just do it for you, which is cool. And so you might say, well, why don't I just have everything all in one big container? Both for Nextflow and just in general. It can be really good to have this type of modularity, the mix and match, because then, for example, if you're having an update in FastQC, it's not going to break some dependency that MOSDEF relies on because they're separate and they're isolated. So you can swap one out and it's not going to affect anything else in your pipeline. So that's quite nice. If you have everything all in one monolithic container, you're starting to edge towards that dependency hell problem that we were trying to get away from in the first place by using containers. So if you want to specify the container that you want your process to use, it's one line that you specify with this. So in your process block, so FastQC, which is kind of what we were looking at before. If I want to use this container, I use this line and I specify where it lives on the internet. So this is the tag. You can also, doesn't have to be from key.io, it can be from Docker Hub. It can also be a local path if you want. So I've been doing some testing also for Bioinformatics actually, so it's protein fold pipeline. And so I've been building my own containers because I'm doing the development and I'm specifying a file where they live on Scratch because I haven't uploaded them to the internet yet because I'm still trying to get them to work. Nextflow cache, so I think we've already talked about this. So it'll pull, Nextflow will pull the file into your cache and it'll live there. And so it doesn't have to pull it every single time it runs, it only pulls it once. And we already talked about bind mounts. But yeah, this is basically how you specify it. And if you want to override the container directive, you can do that yourself. So if you're pulling like a public pipeline and you're like, oh, actually, for me, I need to use this version or this container for whatever reason, you don't have to go into, changing kind of the source code, so to speak, of the workflow. You can put it in a config and then specify that config at runtime, and it'll override the defaults that are in the pipeline. So that's why we were talking about, specifying your own config before, because it allows you to do stuff like this. You can customize things without, having to get into the guts of the pipeline. And so, for example, if I want to update my process BWA mem with this name, I can say, here's the container I want to use actually. And for this guy, I want to use this container. And maybe I have a default for everything else, hypothetically, which would be this one if I wanted to. So you can specify the process like with name. So that's like exactly this process has this name, or with label. So processes can be labeled, for example, like high memory or big job or something like this. And so maybe you have multiple processes with that label, high memory, or in this case, process with GATK. And so you can group all of those to use a certain container, for example. So this is where things start to get a bit complicated. I know it breaks my brain a little bit. So once upon a time, as I said before, you used to have the main.nf and like nextflow.config, and that was kind of it. But pipelines have got more and more complex over time. And so instead of just having one main.nf, you kind of have many. And the primary main.nf imports processes from modules. So this is what the directory structure might look like, right? I've git cloned this example Nextflow, and I can see that I've got my conf, which is short for config, and it contains my various config files. I've got the primary main.nf, but I've also got these modules, and each module also has a little main.nf. And so you have one module per process generally, and each module has its own little main.nf. And these modules get imported to the primary main.nf. So it's a bit like if you're importing something with R or with Python, you know, like from utils import whatever, and Python will go out and find that and import it in at runtime. That's kind of what the main.nf is doing. So rather than having like a 10,000 line main.nf with everything all at once, it says, all right, I'm going to pull in this process, which is stored as module local bolt main.nf, and we'll pull it in at runtime. And the default containers for these modules, for these processes, are defined in the main.nf under the modules, directory structure. So if you're like, well, where is, like, I want to override this default container, but I don't even know where it's specified. Which happened to me recently because I was like, where is this? God, it's been a while. Where are you? That's where they are. So you can have a sticky beat, see what's in there, and decide, do you want to override it or not? And you can override it with a custom config. So you don't have to go in and change this module's local bolts main.nf file. Don't do that. If you don't have to, just use your own custom config. So something else to be aware of is using this latest tag. That is not good for reproducibility because what is the latest tag will change every week, every month, frequently. If you want to be achieving reproducibility, having a tag is good. So this would be having a tag, pinning a version, not bad. The most reproducibility is if you can use the container hash. So it just depends. You can also add container options per process. So we talked before about you can, specify what container you want to use, for example, but you might also want to, include, for example, container process. Like if you want to use NVIDIA GPUs, you're meant to add this --ND flag ahead of your command. And maybe you need to add in like extra bind mounts or something for a particular process. You can also do that here. Something else that can be important. So I talked about the NVE flag for NVIDIA, but you might also have, if you're specifying something to run on a GPU, you might need to tell it, hey, I need you to use this GRES GPU 1, depending on your cluster. You might want to give it the accelerator. So you can do that type of thing as well. So if you are using GPUs with NVIDIA, with Nextflow, just something to keep in mind. if you're using AMD GPUs, you don't need the dash dash NV. There is a dash dash rocking flag, but like you don't really need it, so don't worry about that one. But yeah, for NVIDIA, it does seem to be important. And so, yeah, as I think we talked about, your main Nextflow process will pull the images, put them in a cache, and then all the subtasks will go, oh, okay, the main Nextflow process has already pulled all these containers and I can just use them. But you can have some issues with the pulling of the images. So if you're running your main Nextflow task on a compute node with no internet, it won't work. So you might need to pre-pull the images from like the copy queue if that has internet access. On Photonics, not an issue because everything can speak to the internet. The other thing that does bite me fairly often is pulling these containers can actually be quite memory intensive. And so I've definitely been burned before being like, oh, I'll just specify 4 gigs of memory, no problem. And then my task will run out of memory because it's trying to pull these massive containers and it crashes the task. So if you're going to do that, make sure that you do request enough memory. 36 gigs seems to be a kind of sweet spot for me, but you'll have to see for your own, for whatever you're running. The other thing you can do, which is really handy, is pre-pulling the images. So if you add this preview flag, it will pull the images for you, but it won't launch the pipeline. So if you know, all right, I'm going to pull the images now, specify high memory, run this, and then when you actually run your pipeline for real, you don't need 36 gigs of memory. Just add 2 or 4 or something small because generally that doesn't need a whole lot. It's only for pulling the images. And so here's a real world example of a HPC config. So this is a snippet from, well, it is actually the Pawsey Photonics profile. And so you can see here, Georgie from Biocommons and I have put together this config. So Singularity set up, limiting the number of concurrent jobs with Slurm, just so you're not like spamming the queue too much. I'm also giving it some information about the executor. So on Photonics, we use Slurm. I'm specifying the account. So this is reading from the system environment, your Pawsey project code, and loading the Photonics Singularity module. The cache mode is lenient, which can be important for HPC, so that it doesn't get confused about like, oh, date stamps are a bit off, or just basically gets over a bit of a bug. And you can also do some interesting things where, like, for example, if the task memory is less than a certain amount, it'll go to the work queue. If it's more than that, it'll go to the high mem queue, for example. Yeah, so this is cool if you're working in Photonics. If you're not working in Photonics, though, like a nice example. And most of the big HPC centers will have their own config that you can go find and see what they've set up. All right, so a bit of a recap. We're at the end now. So why containers? Bit of a crash course on how they work. Why would you use Singularity on HPC and how does that kind of work? Integration of Nextflow and containers. Some advanced Nextflow container stuff, which is a bit of a deep dive, and an example working config. So I think that's it. You might have, here's some links. This will be made, the slides will be made available for you so you can look these up later. But otherwise, thank you very much for listening, and I think we're on to questions.
Thank you, Sarah. But we already have some questions that came with the registration forms. Do you pin containers by digest rather than tag? And how do you handle image rot when registries disappear?
Yeah, that's a great question. If at all possible, it is better to pin by hash because it is more specific. And as I sort of said before, you can upload, you can change what is behind the tag. Like you might upload a slightly new recipe or a slightly new container with the same tag. I don't know, I definitely have done that when I realized like, oh, there's a bug in the thing I've uploaded, I better fix it. It's the same tag, but it's a slightly different container. Whereas if you're doing with a hash, that is less likely to happen. With image rot, that can be a problem. it depends where you're getting your containers from. Some will have things there for a long time, others maybe won't. Um, if you have downloaded the image, like downloaded the container, and you have a physical copy of it, you are in control of where that goes. It won't change, and it won't disappear. So if you really, really, really want to be sure, keep that copy locally, and yeah, don't lose it. I guess, unfortunately. So containers are probably one of the best bets we have for reproducibility, but even they aren't completely perfect, unfortunately.
Thank you, Sarah. And then there's a question in the Q&A. I'm building a Nextflow pipeline that uses Obtainer. I'm running into write permission errors when the process attempts to write temporary files inside the container. Do I need to rewrite the code to write temp files into mounted directories, or is this a Nextflow Obtainer setting that can be changed?
Yeah, good question as well. So by default, containers are, excuse me, containers are immutable. So you can't write inside them and change things. Um, so yeah, you wouldn't be able to just write your temp files directly inside the container. That won't work. What you can do is a bit of a hack with the bind mounting. So with this, you can bind mount. So let's say that your process is trying to write to, /temp inside the container, but you actually want it to write to /scratch on your HPC. What you can do is bind mount /scratch :/temp. And so the container thinks it's writing to /temp, but it's actually writing to /scratch. So it's a bit like a symlink. So if it really is insisting on writing to a particular directory, you can use this bind mounting to effectively symlink it to something where it can write. Or you can change the code, up to you. But that is one option.
And we also have a question about the flag profile. Can you specify cloud AWS batch?
Yeah, I mean, cloud computing is something I have to admit I'm less familiar with I work in HPC. Just not something I typically use, but yeah, you can set various flags to make it work for something like that. So I would refer to the documentation and see what's available to make it work on AWS or similar.
And Sarah, I noticed that you've mentioned Conda in your presentation. Can you tell us why would you want to use containers instead of Conda? Don't they both lock down an environment?
Yeah, also a good question. I mean, in general, sometimes Conda does what you need it to do, and that's fine. I'm not anti-Conda as such. However, when you are running your workflow, Nextflow can either pull the container on the fly and reuse it, or it can install this Conda environment, and that I find tends to have more issues. It can be slower, particularly if you're using Conda and not Mamba, which is Mamba's much faster. And I also kind of find that it can be harder to reproduce environments with Conda, like over time. Like we were talking before about like image rot or like, versions shifting away. I find that happens a lot more with Conda than it does with the container. So I do find it a bit less reproducible over time. But yeah, mainly it's the convenient. If you're not overly concerned about reproducibility, then yeah, I just find the containers to be easier and less prone to having issues.
Thank you. And another question we have, Sarah, based on your experience, which is better, Singularity or Docker?
Yeah, so if you're writing your own containers and building them yourself, definitely Docker syntax is better for that, I would say, because it has this like nice caching. So if you get like halfway through all these build steps and then you've done something silly and there's a bug, and you need to fix it and then try again, it'll remember all the cache, like all the layers it's already done successfully. It'll remember those and you don't have to rerun them. So that's really good. Singularity doesn't seem to do that properly or well. And so, that just takes a really long time to build all your images and it's very annoying. So for building, definitely Docker, I would say. For runtime, even if I have sudo access somewhere, like on a VM or whatever, I would still tend to use Singularity as my container engine. So like running my containers because the syntax is a lot nicer, it's a bit friendlier. So yeah, that would be my preference. Docker allows you to do like all sorts of weird and wonderful things that aren't really relevant generally to a bioinformatics type of work. Like, if you're running a big cloud setup and, on a hyperscaler and you're wanting to make sure you've got a lot of redundancy for your website or for Black Friday sales, Docker and Kubernetes, yeah. For bioinformatics, you don't really need that. Singularity, I think, is a nice interface to do what you need to do.
Thank you, Sarah. And let's do one last question. I guess we've talked about containers in Nextflow. What about containers outside of Nextflow? Can we use them?
Yeah, definitely. Use containers like all the time, partly because you don't have to worry about installing stuff yourself. If you're using a pre-built container, someone else has already done all the hard work for you, and you can just pull the container and run the thing generally, which is great. Wish that had been a thing or something I had known about back in like 2019, when I was struggling with VEP because that was driving me absolutely nuts. So container would save all of those issues. You don't need Nextflow to benefit from that. But also, if you're like part of my work at Pawsey has been trying to get these tricky software builds working well and available for users, right? And so with a container, it means I can be very specific about all the dependencies I want in the operating system and like the version of Rockham, which is like the AMD GPU backend type of thing. I can get all of that and lock it down and it's completely isolated. So I don't need to worry about like, oh, well, Onsitonic's X thing isn't available or whatever. I can have my own little sandbox almost with the container, which is like super, super useful. It also gets around issues with like file count. So on a HPC, generally you'll have some kind of like either disk quota and or file count quota. And so if you wanted to install, for example, PyTorch with Conda or Pip, it has so many files and it'll eat up a lot of your quota or maybe even exceed it. If you have it in a container, it just counts as one file. So even though you've got all these really big packages installed inside, the file system just sees one file. And so you can get around the quota issues that way, which can be very useful. So many benefits. Containers are very, very useful.
Thank you, Sarah. Thank you for sharing with us all of this information. Thank you, Sarah, again. Thank you, everyone. This webinar is part of a series of bioinformatics training events organized by Australian Biocommons. You can find out more about upcoming webinars, workshops, and other events on our website. And you can watch recordings of this event and previous events on our YouTube channel. And finally, we'd like to acknowledge our funding. The Australian Biocommons is enabled by and Chris via Bioplatforms Australia. Thanks so much again for watching, and we hope you enjoyed today's presentation and look forward to seeing you again soon. And until next time, goodbye for now.